Good evening, all of you. Am I audible and visible? If I'm audible and visible, can I see a thumbs up on the live chat, please? So, if you're joining my session for the first time, this is Dr. Shanmugapriya. Thank you, Priyanka. Thank you, Karthik. So, this is Dr. Shanmugapriya, Professor and Head of the Department of Biochemistry at Government Thoothukudi Medical College. And I'm your Unacademy educator for biochemistry. And today's session is to discuss all MCQs, which were biochemistry MCQs, which were asked in the INI CET November 2022 that happened today. And as you all know, this session happened in two time slots. One was in the morning and the other one was in the evening. And I've included all biochemistry MCQs of both the sessions. Okay. And the aim of this session is not just for you to know the answers for all these MCQs, but also for you to get ready for NEET PG 2023. Because only when you know what kind of questions are being asked generally, you'll be able to get ready for NEET PG 2023. Okay, so totally I have 20 MCQs. Morning session there were 11 MCQs, and the afternoon session I've received 10 MCQs. And apart from these MCQs, if you can think of any more MCQs which I have missed, please feel free to type it in the live chat. Good evening, Devangan. I am happy. I'm good. Both morning and evening. I have collected questions of both morning and evening session, Indranath. Okay, so before we start the session, let me tell you a few facts about Unacademy's plans. So Unacademy has got now MedMinds Ultra Combat 2.0 exam, which is scheduled for November 27th. And if you're asking me why should I get ready for this exam, because it's a gamified test, and if you participate in this test, you will know at the national level where you stand. And apart from this, yes, it's helpful for FMG, it's helpful for NEET PG 2023 aspirants. Good evening, Amit Kumar. Good evening, all of you. Okay, so about Combat 2.0, it's a gamified test, and the duration of this test would be 60 minutes, and the number of questions that will be asked is 50 questions. And if you're asking me what are the subjects that will be covered, that depends on which exam you're going to take up. So we have unique exams, year-wise exams for MBBS Year 1, Year 2, Year 3. And screen sharing having some problem. Is there a problem? I'm able to see it. Important topic for FMG. After this, I will tell you, Devangan, after I finish discussing all these MCQs. Okay, I don't think there is any issue. Yeah, thank you. So do take this up, because apart from you knowing where you stand, you'll also get attractive prizes. As you can see here, there is a lucky participant. If you're a lucky participant, you might get even MacBook. Okay. And to enroll yourself for this exam, you can use any educator's code. If you want, you can use my code, which is my name, C. Shanmugapriya. So that's about Combat. And we also have FMG All India mock test. So Devangan, you can take it up. It is conducted in two parts, exactly like how you would face your final FMG exam. So part 1 is between 9 and 11:30 a.m. on November 13th, and part 2 is between 2 and 4:30 p.m. on the same day. And we also have NEET PG 2023 Achievers batch, and this will be a rapid revision batch with MCQs. So please try to attend this. It costs you 13,770. It's a nice name. Thank you. I'll tell my parents. Yeah, so this is an ultra rapid revision batch. Here we'll be discussing previous year questions and image based questions. And the duration of this batch would be 4 months. And till now we were having MBBS Prof 1 package in Unacademy. Now we also have launched MBBS Prof 2. So if you're a Prof 2 exam aspirant, you can definitely take this up. Hi PK. Which session was easier? I felt the morning session was easier, because many students told me that the evening session was a lengthy paper. The questions looked as if it was simple, but the choices were quite complex. Yes, I'm a Tamilian. Yes. As I told you, I'm working as professor and head of the Department of Biochemistry at Government Thoothukudi Medical College. Okay. So can we start discussing the morning session? Yours was evening. No worries, PK. You are very well prepared. Okay, so don't worry. So morning session, this is the first question. This was an image based question. How many of you answered it right? So what is that you see in the image? Yeah, in the image, do you see orange tonsils? Good evening, Deepiram. So do you see orange tonsils? Orange tonsils is a feature of what? Tangiers disease. And during the session, I've also given you a shorter way, a shortcut to remember it. Orange tang. How many of you have taken this orange tang? So orange tonsils is Tangiers disease. Okay. So what is, so let's learn few other facts about Tangiers disease also, because as I said, most of you will be NEET PG 2023 aspirants, right? So the one fact that I want you all to know about Tangiers disease is, it is caused by the mutation of ABCA1. ABC stands for ATP binding cassette transporter 1. So ABCA1 mutation causes Tangiers disease. If you want to know what is the role of ABCA1, it helps in collecting cholesterol ester and phospholipids from extrahepatic tissue membranes, and it adds it to HDL. So when that transporter is defective, cholesterol esters, cholesterol and phospholipids will accumulate in extrahepatic tissue membrane, and that causes orange tonsils. And it's not only characterized by orange tonsils, HDL level will be very low in this condition. And there is also hepatosplenomegaly. The other feature of Tangiers disease is hepatosplenomegaly and mononeuritis multiplex. Okay, so that's all about Tangiers disease. Now, for completion's sake, about other choices, old man's disease. What is old man's disease due to? It is because of the defect of acid lipase, lysosomal acid lipase defect. And if at all an image-based question is to be asked on Wolman's disease, they can give you adrenal calcifications. So if you see on an X-ray adrenal calcification, what will be your diagnosis? Your diagnosis should be Wolman's disease. An image like this can be shown. So do you see here? Adrenal calcification is a feature of Wolman's disease. Now what about Gaucher's disease? Which enzyme defect causes Gaucher's disease? I hope you all know it is beta glucosidase defect. And if at all an image-based question is to be asked in Gaucher's disease, they can give you two images. One is tissue paper cells, right? Macrophages which have accumulated glucosyl ceramide will make them look like crumpled tissue paper. That's one image that can be shown. Otherwise, they can show you Erlen-Meyer flask deformity. Yeah, can't you see that in the image here? Erlen-Meyer flask deformity can be shown, and this is a defect that is observed in the distal femur. Yeah, it is observed in the distal femur, wherein there is maximum cortical thinning in the dia epiphysis. Yeah, maximum cortical thinning and absence of concave curvature here. Usually what you expect here is a concave curvature. So if both these are missing, that makes it look like an Erlen-Meyer flask. That is why it is called as Erlen-Meyer flask deformity. That's a feature of which disorder? Gaucher's disease, caused by the defect of which enzyme? Beta glucosidase. A-beta lipoproteinemia. Okay, so if it's A-beta lipoproteinemia, what will you expect? There is going to be low LDL, and that's not going to be characterized by grayish orange tonsils. Okay, thanks Babi Reddy. So if that was one of the choices, then you can easily exclude it. Dark hue blood is due to methemoglobinemia. Dark hue blood. Okay, if blood has got dark hue, it is a feature of methemoglobinemia. Thank you, PK. Happy weekend. Okay, since morning I've been with students getting to know all these questions. It was a happy weekend for me. Thanks, Sarthak. So that's about the first question. Okay, so for the first question, answer is Tangiers disease. Now this is the next question. Glycogen phosphorylase is activated directly or indirectly by? Let me first tell you the answer. The answer for this question is calcium. Yeah, glycogen phosphorylase is activated directly or indirectly by calcium. It's Dr. Doom. Oh, hi Dr. Doom Sarthak. Okay, thanks. Okay, so whenever you're asked a question on enzyme regulation, the first question that you should ask yourself is which pathway does it belong to? So glycogen phosphorylase belongs to which pathway? It belongs to glycogenolysis. Yes, answer is A. Cyclic AMP dependent kinase, you're right. So which pathway does it belong to? It belongs to glycogenolysis. After you know the pathway, try to recollect what are the products of the pathway, because products always inhibit an enzyme. And you know, glycogenolysis gives rise to what are the products? Glucose 6-phosphate and glucose. So both glucose 6-phosphate and glucose being products, they inhibit glycogen phosphorylase. Got it? And then insulin. You know, insulin is a hypoglycemic hormone. A hypoglycemic hormone has got nothing to do with stimulating glycogenolysis, which increases plasma glucose. And we all know glycogenolysis is stimulated by, that's what I'm answering, Dr. Vikram. Insulin is a hypoglycemic hormone. It will stimulate only those enzymes which reduce blood glucose, and we know liver glycogenolysis will increase plasma glucose. So glycogenolysis will be stimulated only by glucagon. Okay, so what is the answer here? Calcium. For those of you who want to know why should calcium stimulate glycogenolysis, because muscle glycogenolysis is dependent on calcium. The calcium that is released from sarcoplasmic reticulum helps in excitation contraction coupling, not only by binding to troponin C, but also by stimulating glycogenolysis. Thereby it facilitates the production of glucose, which will be used through glycolysis for ATP production. And that is why calcium stimulates glycogen phosphorylase. Is that clear? Yeah, this is how you should rule out choices in any question. Don't immediately come to a conclusion. Okay, so the answer here is calcium. Now this is again something which we have, there was no choices insulin, but calcium was there, right? Calcium is the right answer. If you can think of another choice, you can tell me. I will help you exclude that choice also. Okay. So burnt sugar smell of urine, this we have discussed so many times, right? Burnt sugar smell is nothing but maple syrup urine disease. And maple syrup urine disease is caused by the defect of branch chain keto acid dehydrogenase enzyme. And as expected, many of you will get confused between choice C and choice D, because choice C is also related to a branch chain amino acid. But isovaleric CoA dehydrogenase defect causes isovaleric acidemia. What does it cause? It causes isovaleric acidemia, and that is characterized by sweaty feet odor. Yeah, sweaty feet odor. And those of you who have attended my sessions, I would have shown you an image wherein the feet is kept on the ice. Okay, so isovaleric acidemia is caused by, it's characterized by sweaty feet odor. That's not the point, right, Suvo? Suvo, our point is to know the answer and to know how to exclude other choices. I intentionally gave you all these choices because all these choices are related to some smell of the urine. But we know the answer is branch chain keto acid dehydrogenase. Is that clear, Bhattacharya? Yeah, isovaleric acidemia is characterized by sweaty feet odor. Yes, I've taken sessions on mucopolysaccharides, Sarthak. Okay, so this is not isovaleric acidemia. Fumarate acetoacetate hydrolase defect causes which disorder? Fumarate acetoacetate hydrolase defect causes type 1 tyrosinemia. Okay, so let me give you all answers now. So what is answer here? It is branch chain keto acid dehydrogenase. Now phenylalanine hydroxylase defect causes what? The condition is phenylketonuria. And phenylketonuria is characterized by what odor of the urine? Mousy odor of the urine. Fumarate acetoacetate hydrolase defect, very good, causes type 1 tyrosinemia. Type 1 tyrosinemia is characterized by what? It is characterized by cabbage odor. Branch chain oxidase. Okay, it is not branch chain oxidase, it is branch chain keto acid dehydrogenase. Okay. And then isovaleric CoA dehydrogenase defect is isovaleric acidemia, and this isovaleric acidemia is characterized by sweaty feet odor. Yes, risk of hepatocellular carcinoma is very high in type 1 tyrosinemia. Very good, Divya. Now this is another question, interesting question. This also we have discussed in the session so many times. Cystic fibrosis is caused by which of the following deletion? It is caused by the deletion of phenylalanine at which position? At 508 position. Good, Sarthak. Boiled cabbage odor is a feature of hypermethionemia. I'm very happy that all of you are interacting in the live chat. It makes the session really interesting. So answer is phenylalanine at 508 position is caused by a deletion mutation. But otherwise, what are the facts that you need to know? You knew only F508. Good. Good for you. Okay, so the fact about cystic fibrosis, it's an autosomal recessive condition and is caused by the defect of which protein? The protein that is defective is CFTR protein. What is CFTR protein? Cystic fibrosis transmembrane regulatory protein. Okay, and what is this protein related to? This protein is related to movement of halide. It is related to movement of halide ions across the ducts, which are related to secretion of sweat, related to secretion of digestive juices, as well as mucus. It is related to halide movement, wherein they are related to halide movement of the ducts related to glycogen phosphorylase cyclic AMP option was also there. Okay. If there was cyclic AMP can activate glycogen phosphorylase for sure. Okay, cyclic AMP dependent protein kinase will stimulate glycogen phosphorylase. But if the question was muscle glycogen phosphorylase, the answer will be calcium. Do you understand that? Okay, so it's related to secretion of sweat, digestive juices, as well as mucus. And in sweat, as you all know, it is related to releasing chloride ion. Where does it release chloride ion? It releases chloride ion into the cell from the duct. So when that transporter is defective, excess chloride will be secreted in the sweat. So what is one of the diagnostic investigations for cystic fibrosis? Sweat chloride concentration will be higher. Yeah, in this condition, one of the features is sweat chloride concentration. The names and abbreviations of amino acids. Maybe I'll have a session separately for amino acid chemistry, Sarthak. Yeah, it will take a longer time. For example, tyrosine is Y. It will take a longer time. Yeah, so what was I telling you? Sweat chloride concentration will be higher in cystic fibrosis. Okay. And in all other places, what does the transmembrane regulator do? It causes chloride ion to enter into the cell. Do you get it? So this is about cystic fibrosis, and there are around 700 mutations causative of cystic fibrosis, but the most common mutation which results in cystic fibrosis is caused by a deletion mutation, wherein 3 nucleotides are lost, and that causes a deletion of phenylalanine at 508 position. Okay. Now the significance of HMP shunt are all except. Does it act as a source of NADPH? Does it act as a source of ribose 5-phosphate, source of acetyl-CoA? Does it utilize ribose? Were these the choices provided, all of you? The significance of HMP shunt are all except. Yeah, answer is utilizes ribose. HMP shunt does not utilize ribose. It acts as a source of ribose 5-phosphate. Very good, Chetan. Very good, Amit Kumar. So HMP shunt, I would like to tell you that there are 2 significances of HMP shunt. One is that it acts as a source of NADPH. The other one is that it acts as a source of ribose 5-phosphate. And that is why HMP shunt is otherwise called as pentose phosphate pathway. And what is the use of ribose 5-phosphate? It is used for purine and pyrimidine nucleotide synthesis. Okay. And does it use... B and D are contradictory. Yes. Yeah, just by that fact you could have chosen the right answer. You're right. Okay, so purine and pyrimidine nucleotide synthesis. So we know it acts as a source of NADPH. We know it acts as a source of ribose 5-phosphate. Now when you come to source of acetyl-CoA utilizes ribose, we know for sure it does not use ribose. What it takes is it takes glucose 6-phosphate. This glucose 6-phosphate goes through 2 steps, wherein it loses 1 carbon dioxide, and that is how a hexose glucose 6-phosphate becomes a pentose, which is ribulose 5-phosphate. Then this ribulose 5-phosphate directly becomes ribose 5-phosphate. It does not use ribose for synthesizing ribose 5-phosphate. Ribulose 5-phosphate will become ribose 5-phosphate. Yes, morning session was easy. You're right, PK. Okay, so it does not utilize ribose. Now, does it act as a source of acetyl-CoA? Yes, HMP shunt can act as a source of acetyl-CoA indirectly, because in HMP shunt, when glucose 6-phosphate molecules enter into the pathway, they come out as 2.5 glucose 6-phosphate. Yeah, 3 glucose 6-phosphate will be used, and they come out as 2.5 glucose 6-phosphate, and the half glucose 6-phosphate is nothing but glyceraldehyde 3-phosphate. And this glyceraldehyde 3-phosphate gets into glycolysis. What will you get as a result of glycolysis? You get pyruvate. and this pyruvate gets converted to acetyl-CoA. So indirectly it can act as a source of acetyl-CoA. Is that clear, Divya? Yeah, even before you could ask, I've told you the answer. So the significance of HMP shunt is that it acts as a source of NADPH, it acts as a source of ribose 5-phosphate. Indirectly, it acts as a source of acetyl-CoA because it comes out as 2.5 glucose 6-phosphate molecules, and that will get into glycolysis, form pyruvate, and that will form acetyl-CoA. Okay. Now the next question. Refsum's disease is caused by the defect of. So in the morning, Refsum's disease was asked, and in the evening, Zellweger syndrome was asked. And what is the common point between Refsum's disease and Zellweger syndrome? Both are related to peroxisome. Yeah, both are related to peroxisome. So it's caused by the defect of, Dr. Vikram is right. So it's caused by the defect of alpha oxidation of branch chain fatty acids. Yeah, somewhat they were related, because both in the morning and in the evening I heard that there was a question on oxyhemoglobin dissociation curve. Okay, so Refsum's disease is caused by the defect of alpha oxidation of branch chain fatty acids. And if you want to know more about Refsum's disease, the presentation will be close to the adolescence period. It is not going to present in the neonatal period or in the infantile period. Presentation is in the adolescence time, and around that point they would present with cerebellar degeneration symptoms. They present with ataxia and ichthyosis, and it's caused by the defect of phytanic acid hydroxylase. The enzyme that is defective here is phytanic acid hydroxylase, and phytanic acid is a branch chain fatty acid that gets metabolized only with the help of peroxisome. Got it? Yeah. SARS-CoV-2 diagnosis is done by all except. Phytanic acid oxidase is also right. Hydroxylase is also right. Hemoglobin dissociation curve is not there in the evening. Okay. So SARS-CoV-2 diagnosis is done by all except. What do you think is the right answer? I know that first point that you should recollect about SARS-CoV-2 diagnosis is, do you all know that SARS-CoV-2 is an RNA virus? Yeah, SARS-CoV-2 is an RNA virus. So what should you do to measure RNA? First, you should convert RNA to DNA. For this, you will employ reverse transcription step. So that reverse transcription PCR is called as RT-PCR. After reverse transcription, you quantify it using real-time PCR. But if you don't want complex diagnostic technique, you can also estimate the antigen that is present in SARS-CoV-2 using immunoassays. Okay, so 6 is not that question. So what was the question, Prasad? Please feel free to type it. Yeah, everything sounds F. Okay, that's a good way of remembering it. So what is the right answer here? The right answer here is Southern blotting. Southern blotting is not used for SARS-CoV-2 diagnosis because Southern blotting detects DNA. Don't we all know that? Real-time PCR against M protein. A bit different but same gist. Okay. Refsum's disease is related to peroxisome, Prasad, for sure. So let me tell you few facts about peroxisome. Yeah, peroxisome is related to just 3 functions. Please memorize these 3 functions. The first function is it's related to very long chain fatty acid oxidation. Can you memorize it? Yeah, peroxisome is related to very long chain. Southern blotting is for DNA. Yeah, northern blotting is for RNA. Right. South is dosai. Oh, that's a good way of remembering it. So it's related to very long chain fatty acid oxidation. Second one is it's related to alpha oxidation of branch chain fatty acid. Third function is plasmalogen synthesis. This can also be asked as an MCQ in your NEET PG. So what are the functions of peroxisome, all of you? Peroxisome is related to very long chain fatty acid oxidation, branch chain fatty acid oxidation, and plasmalogen synthesis. So whenever there is a peroxisome defect, you will have to anticipate all these defects. Okay, good. So here the answer is Southern blotting. Southern blotting is used only to detect DNA, and your SARS-CoV-2 virus is RNA. Okay. Now, selenocysteine is present in plasmalogen. If you want to know what is a plasmalogen, Divya is curious about plasmalogen. Plasmalogen are glycerophospholipids with ether linkage. Yeah, plasmalogen. She wants to know what is a plasmalogen. It's a glycerophospholipid with ether linkage. Do you want to know more about it? I don't think it'll be asked to you. It's just a phospholipid. If you know that, that's enough. Okay. Selenocysteine is present in which of the following enzymes? What is your answer? It is related to glutathione peroxidase. Selenocysteine is present in glutathione peroxidase. So all of you, you might get confused here. As far as glutathione reductase is concerned, glutathione reductase uses riboflavin. I think I've given you a clue to remember this. Ribbon and glue go hand in hand. Yeah, ribbon and glue go hand in hand. So ribbon is used for glutathione reductase. Glutathione peroxidase needs selenocysteine. Other enzyme, idolase. Idolase does not need selenocysteine. Okay, so I will tell you what are the enzymes which are dependent on selenocysteine's presence. The first one is glutathione peroxidase. The second one is thioredoxin reductase. Yeah, second one is thioredoxin reductase, and the third one is 5'-deiodinase. Western blot is used for protein detection. I can't read your name. I think it's imasic. Yeah, Western blot is for protein detection. So what are the three enzymes which are dependent on cell? Other choices were glutathione peroxidase, glutathione synthase, deiodinase, thioredoxin reductase. In that case, maybe they missed except. It should have been except, because glutathione peroxidase, deiodinase, thioredoxin reductase, all these need selenocysteine. Only glutathione synthetase or synthase will not need selenocysteine. Okay. Maybe they missed except. It was multiple choice. Oh, if it was multiple choice, then these three should be the right answers. Okay, multiple answers, these three. 5'-deiodinase definitely needs selenocysteine. Okay. Yeah, then answer is glutathione reductase. True about sirtuins except. So how many of you are interested to know about sirtuins? Yeah, how many of you want to know about sirtuins? It's something which is interesting, right? Because it is supposed to delay aging. So let me tell you all that you need to know about sirtuins first. Okay, so then you will be able to choose the right answer. So sirtuin was first observed in yeast, and it's an abbreviation of something called a silent mating type information regulatory protein. Yeah, it is silent mating type. You all like to know that it's present in red wine, right? Yes, it is present in red wine. So it's related to silent mating type information regulatory protein. Function wise, what does it do? It is one protein which simultaneously catalyzes two enzymatic activities. The first activity is ADP ribosyl transferase. This was in pathology. Okay, so it's always cross dimensional, right? So everything is related one way or the other. So what is it related to? It has got two enzymatic activities. The first enzymatic activity is ADP ribosyl transferase. And the next one is histone deacetylase. So I'm typing, I've typed everything because that will help you in visual memory. Memorize it. It has got two enzymatic activities. One is ADP ribosyl transferase. The other one is histone deacetylase. So whenever there is histone deacetylation, the chromatin will undergo condensation. Because when you remove acetyl groups from histones will be positively charged and they interact with DNA and DNA will be condensed. So what will it result in? It causes chromatin condensation. Can't tell the difference now. Yeah, everything is integrated, right? So it results in chromatin condensation. And initially what we thought sirtuin only suppresses transcription, because whenever chromatin gets condensed, there can be no replication and no transcription. We thought sirtuin is a transcription repressor. But later we found out that by chromatin condensation, it helps in DNA repair mechanism. Don't you think it's interesting? It helps in DNA repair mechanism. The most common DNA error that is corrected by sirtuin is base excision error. Yeah, base excision error can be easily corrected by sirtuins. And now do you understand why or how it delays aging? Via how it delays aging? It delays aging because it helps in DNA repair mechanisms. Okay, and additionally what I would want you to know is So once we know that, once we understood that it delays aging, we started doing so many experiments on sirtuins, and that is when we identified that there are 7 types of sirtuins in human being, out of which T1 and T6, both of them are related to glucose balance. So what they do is they improve glucose tolerance, they improve glucose metabolism. Yeah, they also help in glucose metabolism. So having understood all these, can you start excluding choices one by one? Sirtuins, do they improve insulin sensitivity? Yes, they do that. And is it a histone deacetylase? Yes, it's a histone deacetylase, thereby it causes chromatin condensation. Does it increase the lifespan? Yes, it increases the lifespan and it delays aging. Okay, now about the last one, is it stimulated by niacinamide? For answering this statement, for answering about this statement, I want you to know that this ADP ribosyl transferase uses NAD as its coenzyme. So what does it use as its coenzyme? It uses NAD as its coenzyme, which then gets converted to niacinamide. So niacinamide is a product of sirtuin, and I told you products always inhibit an enzyme. So niacinamide is an inhibitor of sirtuin. So whenever you're trying to anything to do with telomerase, no, it has got nothing to do with telomerase activity. That's a totally different way of improving or increasing your lifespan. This is totally different. Okay, so niacinamide is an inhibitor of, it was not in the question. Okay, what was the option? Yeah, they had different set of questions, MedStudy blog. What was another option? It has 7 proteins. Yes, it has 7 proteins. Okay, so niacinamide inhibits sirtuin. And interestingly, when NAD is necessary as a coenzyme, what will happen when a person is on a high calorie diet? When you're on a high calorie diet, your NADH level will be high. Or in other words, all NAD will get converted to NADH. And when there is NAD deficiency, sirtuin will become inactive. When there is low sirtuin, there will be accelerated aging. Now do you understand why we always say, now do you understand why we always say high calorie diet accelerates aging? Because when you're on a high calorie diet, what will happen? All NAD will get converted to NADH. NAD is essential for sirtuin. Without sirtuin, there is accelerated aging. And this is why fasting, how many of you have followed intermittent fasting? Intermittent fasting delays aging. How does it delay aging? Because NAD level will be high, sirtuin will be activated. Do you understand this? High NADH suppresses sirtuin action. That is why high calorie diet causes accelerated aging and fasting delays aging. Okay. So are you clear about sirtuins? If you want me to repeat it, I can repeat it. So what are the facts that you have learned about sirtuins? Sirtuins are nothing but silent mating type information regulatory protein. And what is their main function? They have 2 enzymatic activities. One enzymatic activity is it is an ADP ribosyl transferase. And the second enzymatic activity is histone deacetylase. What does it achieve by acting as histone deacetylase? It causes chromatin condensation. It helps in DNA repair mechanism. Because it causes DNA repair, it delays aging. And then we started analyzing all these sirtuins and we understood that sirtuins also help in glucose balance and glucose homeostasis. And for this activity, they are dependent on NAD. And anything which decreases NAD level will accelerate aging. That is why high calorie diet accelerates aging and fasting will delay the aging process. Can you explain option number A? I told you, right? Sirtuin 1 and 7. So I know I'm blocking it. So can you see that here? Sirtuin 1 and 6 help in glucose homeostasis. It increases insulin secretion. It improves insulin sensitivity. So true about sirtuin except it is stimulated by niacinamide. If you can think of any other choices, I'll be very thankful to you. So did you see this question, all of you? Choose the right sequence of steps in the conversion of cholesterol to testosterone. Yes, I would say this is a very complex question because remembering this sequence of steps is difficult unless you have been learning it multiple times. So let me tell you how to remember all this. So the first fact that you should know, I'll first tell you the right answer here. The right answer here is choice A. Yeah, 2022 desmolase first, followed by 3 beta hydroxysteroid dehydrogenase, and then 17 alpha hydroxylase, and finally it is 1720 lyase. The first enzyme is cholesterol desmolase. So I'm going to use this question to tell you few other facts, like why do we call progesterone as a C21 steroid? Why do we call androgens as C19 steroids? Why do we call estrogens as C18 steroids? Yeah, can we learn about it now? Yeah, true color of INI-CET. Yeah, don't take it to heart, Sarthak. Our aim is to know new facts, to know new concepts, okay? So, had you learned all the subjects conceptually, you would have excluded most of the choices, you would have given the right answer. But ultimately, if it's a pure fact-based question, don't be harsh on yourself. Nobody can answer it. Okay? Nobody can answer it. So don't worry about it. So I'm going to tell you about steps involved in the conversion of cholesterol to testosterone. So the first step is you take cholesterol, and this cholesterol is a C27 steroid, in the sense it has got totally 27 carbon atoms. And this cholesterol is acted upon by the first enzyme, which is cholesterol desmolase. Yeah, that was the first enzyme which I showed you. The first enzyme to act on cholesterol is cholesterol desmolase. And this desmolase removes 6 carbon atoms, and it converts cholesterol into pregnenolone. So all of you tell me, what kind of steroid is this pregnenolone? Pregnenolone is a C21 steroid. And this pregnenolone is then acted upon by the next enzyme, 3 beta hydroxysteroid dehydrogenase. The next enzyme is 3 beta hydroxysteroid dehydrogenase, which converts pregnenolone to progesterone. So now tell me, progesterone is what kind of a steroid? Progesterone is a C21 steroid. I haven't even given the exam and feeling so nauseated seeing such questions. God bless my seniors. No, Sarthak, what I would say is, only when you know the sequence of steps involved in the conversion of cholesterol to testosterone, you will be able to understand congenital adrenal hyperplasias. How many of you have found it difficult to understand congenital adrenal hyperplasias from a medicine textbook? You think it's tough. Why do you think it's tough? Only because you don't know the sequence of steps involved in the conversion. But once you know the sequence of steps, you will understand what happens when there is a defect at various places. So probably I'll do a YouTube session on congenital adrenal hyperplasias. If you watch that session, you'll understand the significance of this question. INI-CET, they don't ask you a question for no reason. Yeah, why have they asked you this? Because they know only when you know the sequence of steps, you'll be able to understand congenital adrenal hyperplasias. Okay? Yeah. So progesterone is a C21 steroid. The next enzyme is 17 alpha hydroxylase. And 17 alpha hydroxylase converts progesterone into 17 hydroxyprogesterone. And the next enzyme is 1720 lyase. And 1720 lyase removes 2 carbon atoms, and that is how 17 hydroxyprogesterone gets converted to androstenedione. Have you heard of androstenedione? This androstenedione then gets converted to testosterone, and that becomes dihydroxytestosterone. From C21 steroid, because you have removed 2 carbon atoms, androstenedione, testosterone, dihydroxytestosterone, all these are C19 steroids. And how will you get estrogen from androgen? When aromatase acts on androgen, removes 1 carbon atom, you get estrogens. So all estrogens, estrone, estradiol, estriol, they are all C18 steroids. Is that clear, all of you? These are all C18 steroids. So this is the sequence of steps. So I'm going to show you this in a slide. So first step is cholesterol. What is the first enzyme? The moment I start saying biochemistry, core biochemistry, students start disappearing. But I would like to stress this fact: if you don't understand core biochemistry, you will not understand clinical biochemistry. Okay, so what is the first enzyme? Cholesterol desmolase acts on cholesterol, and it removes how many carbon atoms? 6 carbon atoms, and it converts cholesterol into pregnenolone. So pregnenolone is a C21 steroid. And then the next enzyme is 3 beta hydroxysteroid dehydrogenase, which converts pregnenolone to progesterone. So progesterone is a C21 steroid. Now progesterone is acted upon by 17 alpha hydroxylase, and it is converted to 17 hydroxyprogesterone. And then 1720 lyase converts 17-hydroxyprogesterone into androstenedione. And this androstenedione becomes testosterone and hydroxytestosterone. Okay, so what is the right answer here? The right answer here is desmolase, 3-beta hydroxysteroid dehydrogenase, 17-alpha hydroxylase and 1720 lyase. So apart from this, what is the take-home message I've told you? C21 steroids include what? C21 steroid includes progesterone number 1 and then your aldosterone cortisol. Yeah, progesterone, aldosterone, cortisol, all these are C21 steroids. What are the C19 steroids? All male sex hormones, DHEA, yeah, DHEA, androstenedione, testosterone and dihydroxytestosterone, all these are C19 steroids. What are the C18 steroids? All estrogens are C18 steroids, okay. So did you see this question? 2,3-BPG causes unloading of oxygen to tissues because of what? After learning 10 times, you forget it again. Yeah, there is no secret behind memory. It's just repetition. If 10 times, if you read it 10 times and then you keep forgetting, it means you have to increase the number of times you learn. Okay, but once you start applying it, you will not forget it. Okay, Sumitra? Yeah, what is the right answer? 2,3-BPG causes unloading of oxygen to tissues because 2,3-BPG has got nothing to do with ATP production. What is 2,3-BPG? It's an intermediate of glycolysis. That's not going to take part in ATP production at all. So it's neither choice A nor choice B. But what does it do? We all know 2,3-BPG reduces the affinity. Very good, Chetan. I'm happy that you remember. So 2,3-BPG reduces the affinity of hemoglobin for oxygen. It helps in unloading oxygen to tissues because it stabilizes something called a STOT structure. It stabilizes something called a STOT structure by favoring salt bridge formation. So answer is choice C. But for you to understand about choice C, let me tell you few facts. Yeah, just be with me for 5 minutes. You will understand everything. So what is hemoglobin structure? Every hemoglobin has got heme in the center. Yeah, I've taught this in hemoglobin myoglobin session multiple times. So hemoglobin has got 4 heme and every heme will be surrounded by 1 globin chain. And because it has got more than 1 polypeptide chain, it can exhibit 2 structures. One is a taut structure, the other one is a relaxed structure. Okay, taut structure means there is strong interaction among the individual globin chains. When salt bridges are formed between individual globin chains, that's called as taut structure. When there is weak interaction among individual globin chains, that is called as relaxed structure. And taut structure favors deoxygenation. As who answered it? As Chetan has answered it. I always say this. Taut structure is like you squeezing out oxygen. Yeah, taut structure is like you squeezing out oxygen. So taut structure favors deoxygenation and relaxed structure favors oxygenation. Are you clear about it so far? Okay. Now what does 2,3-BPG do? When you have such 4 globin chains, yeah, when you have 4 globin chains that are placed close to each other like this, do you see that it encloses a diamond shaped cavity in the center, all of you? Yeah, when 4 globin chains are placed close to each other, it encloses a diamond shaped cavity in the center. And this diamond shaped cavity is surrounded by positively charged amino acids like histidine and lysine. And how about the charges of 2,3-BPG? Can anybody answer this? How about the charges of 2,3-BPG? 2,3-bisphosphoglycerate. Phosphate provides negative charges, glycerate provides negative charges. Okay, so 2,3-BPG is negatively charged. So one molecule of 2,3-BPG sits in the central cavity and it forms salt bridges because it's negatively charged. It interacts with positively charged amino acids nearby. So that favors salt bridge formation. And this salt bridge formation favors what structure? Salt bridge formation favors taut structure. And taut structure favors what? Deoxygenation. Do you understand this? That is how 2,3-BPG helps in unloading oxygen to tissues. Have you understood this? So anytime you're asked about this, you should know this. So what are the facts that I've told you? In hemoglobin, there are two structures. One is taut structure, the other one is relaxed structure. Taut structure favors deoxygenation, relaxed structure favors oxygenation. 2,3-BPG sits in the central cavity, which is surrounded by positively charged amino acids. It favors Sol bridge formation, that favors taut structure. Easy, moderate or hard, that depends upon how you are prepared for this question, for this topic, Divya. If you ask me, I would say it is easy because I have taught everything conceptually. Okay, so however twisted the question is asked, you will be able to answer if you learn it conceptually. Okay, so read it. It has got nothing to do with ATP production. What does it? It binds to the central cavity of hemoglobin, surrounded by histidine and lysine, and it favors Sol bridge formation. Okay, the right answer here is choice C. Does it inhibit taut structure? No, it favors taut structure. Wrong ho gaya. It's okay, Divya. It's all a learning curve. You will be able to answer when such a question is asked in NEET PG 2023. And this is how INI CET questions can be anticipated. Okay. So identify the left curve. I was told that this is exactly the curve that was given in the exam, right? The made questions taught. Yeah. Identify the left curve given the fact that the right curve is of adult hemoglobin. So the right curve, the blue curve is sigmoidal. Don't you see that it's a sigmoidal curve? Whereas the left curve, it is hyperbolic. According to you, what was the standard of paper? I would say it's a moderate paper. It's neither too easy nor too tough. There were many factual questions which you'll be able to answer, but there are some conceptual questions like what we discussed previously. So it's a moderate paper. If you had read it conceptually, I think you'll be able to get good rank. Yeah, what is the right answer? This is a hyperbolic curve. Hyperbolic curve is of myoglobin. Okay. So that's all about morning session questions. Now about evening session questions. Yeah, this is the first question that I came across. Curve shifted to the right. Was that a question? If the curve is shifted, yeah. The right answer is assertion and reason are true, but reason is not the correct explanation for assertion. Because phenylketonuria is characterized by mousy odor that all of us know. Yeah, it is characterized by mousy odor. And fetal hemoglobin shifts the curve to the left, Divya. But the fetal hemoglobin curve will also be sigmoidal. If a sigmoidal curve is shifted to the left, then that can be fetal hemoglobin or it can be carbon monoxide poisoning. But the curve that was shown there is hyperbolic curve. If it's a hyperbolic curve, it is myoglobin. Yes, so many questions. Yes, Sartak. How many should get right, ma'am, out of 11 questions to stay in race? Because it's not just about biochemistry, right? Had you gotten at least 6 right, I think you are in the right path out of 11. If you have gotten 6 right, you are in the right path. Okay. Yeah. So what is the reason for this mousy odor? Because in phenylketonuria, there is phenyl acetate which accumulates. Okay, that phenyl acetate is the reason for mousy odor. So assertion is true. Now look at the reason. Tyrosine and norepinephrine are not formed in phenylketonuria. That is also the right statement, right? This is also a right statement because in phenylketonuria, when phenylalanine hydroxylase is defective. Yeah, phenylketonuria is caused by the defect of phenylalanine hydroxylase, wherein phenylalanine will not be converted to tyrosine. Without tyrosine, what cannot be synthesized? No norepinephrine, no dopamine, no epinephrine. So, of course, in phenylketonuria, there is low norepinephrine, epinephrine and dopamine, but that's not the reason for the mousy odor. So, what will be the right answer? The right answer is both assertion and reason are true, but reason is not the correct explanation for assertion because what is the correct explanation for assertion? It is phenyl acetate accumulation. So, answer is choice B. Zellweger syndrome is caused by the defect of which of the following organelles? What did I tell you? Zellweger syndrome is caused. What is the other name for Zellweger syndrome? It is cerebral hepatorenal syndrome. Yeah, it is cerebral hepatorenal syndrome and it is one of the 3 peroxisomal biogenesis disorder. Here it's not a defect of an enzyme. It is caused by a defect of peroxisomal biogenesis. Peroxisome formation by itself is defective. And what is the function of peroxisome? I told you previously. Can you all recollect and tell me? Yeah, what is the question of Prasad? Prasad, it is left side deviation of the curve. Yes, Prasad, left side. So all of you listen to this. Many of you have a doubt in that question. Left side shift of oxyhemoglobin dissociation curve. What are the causes? It can be because of alkalosis. Because acidosis shifts the curve to the right, alkalosis will shift the curve to the left. That's one cause. Otherwise, it's also caused by fetal hemoglobin. Because fetal hemoglobin exhibits very high affinity, it shifts the curve to the left. Or it can be because of carbon monoxide poisoning. But in the case of alkalosis, fetal hemoglobin and carbon monoxide poisoning, the curve will be sigmoidal. Yeah, but what was the curve which I showed you? The curve was hyperbolic. Okay, that is why I chose myoglobin. Does that answer your question, Prasad? Fetal hemoglobin, you want me to explain? How many of you want me to talk about fetal hemoglobin now? I can tell you. Yeah, I can tell you in detail about fetal hemoglobin. So what is the difference between adult hemoglobin and fetal hemoglobin? Adult hemoglobin has got alpha 2, beta 2, right? Yeah, adult hemoglobin has got alpha 2, beta 2. Whereas fetal hemoglobin has got alpha 2, gamma 2. So what is the difference between beta chain of adult hemoglobin and gamma chain of fetal hemoglobin? Gamma chains lack the positively charged amino acids about which I was telling you previously. So when there is not many positive charges here, will 2,3-BPG be able to bind here? All of you answer this. I said 2,3-BPG comes and binds to the central cavity only because of these positive charges. Those positive charges are present only in beta chain, not present in gamma chain. So there is no positive charge. So there is low affinity for 2,3-BPG. When there is low affinity for 2,3-BPG, what will happen to affinity for oxygen? Affinity for oxygen increases. So what is the difference between adult hemoglobin and fetal hemoglobin? Fetal hemoglobin exhibits very high affinity for oxygen. And when the affinity for oxygen increases, the curve gets shifted to the left. Do you all understand that? Yeah, the curve gets shifted to the left. So curve getting shifted to the left. Can you all tell me the causes? Curve getting shifted to the left can be because of alkalosis, number 1. Number 2, carbon monoxide poisoning. Number 3 is fetal hemoglobin. But in all these cases, how will the curve look? The curve will look sigmoidal. But the curve that was shown in that image was hyperbolic. That is why it is myoglobin. Got it, all of you? Okay, so coming back to this, I was telling you about peroxisome. I said peroxisome is related to very long chain fatty acid oxidation. It is related to branch chain fatty acid oxidation and plasmalogen synthesis. So Zellweger syndrome is characterized by a defect of all these three, and that is why it presents as cerebral hepatorenal syndrome. It is one of the 3 peroxisomal biogenesis disorder. So what is the right answer here? Which organelle defect causes Zellweger syndrome? It is peroxisome. Long-term glycemic control in a person with hemolysis is assessed by estimating fructosamine. All of you tell me, if you want to assess long-term glycemic control, what is the parameter that you will estimate? Long-term glycemic control, we are going to estimate HbA1c. Right? Yeah, infantile refsems. It's not any refsems, it is infantile refsems. And what is your doubt? Okay, so long-term glycemic control is assessed by HbA1c estimation. But what is that you know about HbA1c? It is glycated hemoglobin. So if a person is suffering from hemolysis, can you estimate glycated hemoglobin? No. In a person with hemolysis, there will be rapid turnover of RBCs. So hemoglobin lifespan will be reduced. If hemoglobin lifespan is reduced, HbA1c will also be falsely low. Do you understand this? So you cannot estimate HbA1c in a patient with hemolysis. So what are you going to estimate? You're going to estimate fructosamine. What is fructosamine? It is glycated plasma proteins. Fructosamine is nothing but glycated plasma proteins. Major plasma protein is albumin. That is why fructosamine is otherwise called as glycated albumin. So what is the right answer here? Fructosamine is a protein. It is not a glycosaminoglycan. It is not urea. It is not fructose. Got it, all of you? So if the patient has got a different turnover of RBCs, for example, hemolysis or iron deficiency anemia, in all these cases, do not estimate HbA1c because it will be falsely low or falsely high, in which case you'll have to estimate fructosamine, which is nothing but glycated albumin, glycated plasma protein. Which of the following tissues are dependent on insulin for glucose uptake? Was this a multiple choice question? Yeah, was this a multiple choice question? I hope it was that. Because insulin dependent glucose uptake happens in, insulin dependent glucose uptake happens, I will tell you. Yeah, insulin dependent glucose uptake happens through GLUT4 transporters, and these GLUT4 transporters are present in skeletal muscle, yeah, cardiac muscle and adipose tissue. GLUT4 transporters are present in skeletal muscle, cardiac muscle and adipose tissue. And insulin dependent glucose uptake also happens in hepatocytes. I've told you this so many times. It also happens in hepatocytes, though not directly, but because it stimulates glucokinase. Okay, was it a multiple choice question? No. In that case, I think you would be given mark irrespective of whether you choose it as muscle or adipose tissue, because both are dependent on insulin 100%. Skeletal muscle, cardiac muscle, smooth muscle, all these three are dependent on insulin. Adipose tissue is also dependent on insulin. Okay, brain. Brain is not dependent on insulin. This is for sure not the right answer. Okay. Another option was pancreas. Okay, pancreas glucose uptake is not dependent on insulin. So we can exclude choice A and choice D. Between muscle and adipose tissue, I would say both are right. Okay. Okay, this is an interesting question, right? They are expecting you now to read all previous incidents or accidents, health-related accidents. So, Hooch tragedy is related to which of the following alcohols? Hooch is nothing but a spurious alcohol, right? It is nothing but spurious alcohol, and spurious alcohol tragedy is mainly related to methanol poisoning. Okay, so during distillation, sometimes methanol can get generated. So if it's a distilled alcohol, we remove all this methanol and other alcohols. We see to it that it has got only ethanol. So Hooch tragedy is related to methanol poisoning. Why is methanol toxic? Because when methanol is metabolized by alcohol dehydrogenase, it becomes formaldehyde. And this formaldehyde then becomes formic acid. And this formic acid damages optic nerve, and that is why it causes blindness as early as 12 hours. And apart from this formic acid also inhibits electron transport chain complexes. Particularly it inhibits cytochrome C oxidase. Okay, formic acid inhibits cytochrome C oxidase, and when electron transport chain is inhibited, aerobic metabolism is affected. When aerobic metabolism is affected, that will present as histotoxic hypoxia. Yeah, what will it present as? It presents as histotoxic hypoxia. Is this a new question? Is this a new question? We have not seen this question before. So Hooch is a spurious alcohol. You are worried about spurious alcohol because methanol poisoning is highly probable. Why are you worried about methanol poisoning? Because methanol gets metabolized to formaldehyde and formic acid, and formic acid damages optic nerve, that causes optic neuropathy and blindness. And and apart from that, formic acid also inhibits cytochrome C oxidase of electron transport chain. So it presents as histotoxic hypoxia. Okay. So answer here is choice A, methanol poisoning. Which of the following is true? It's a very simple question, I would say. Yeah, in contrast to the last INI CET exam, the last INI CET, there were so many questions in molecular biology, and that too there were complex questions. But this time there are very less number of molecular biology questions. I think this is the only question that there was one other question. So which of the following is true? To break AT bonds, high temperature is required. No. So AT, there are 2 hydrogen bonds. GC, there are 3 hydrogen bonds. So if there are more AT sequences, TM will be less. If there are more GC sequences, TM will be high. So what is the right answer? To break AT bonds, low TM is required. Okay, Chetan, why did you choose a wrong answer? Yeah, Chetan is right. Answer is C. Gunda Ramakrishna is not right. Okay. So Ramakrishna, answer is choice C. To break AT bonds, low TM is required. And this question was asked. PK told me that there was an image based question on non-competitive inhibition. Did you all see this question? Yeah, so this graph, what is the observation from the graph? So in the graph, do you see that this is an uninhibited curve? Decrease to 3 bpg. Yeah, so this is an uninhibited curve, this is an inhibited curve. And irrespective of whether it is uninhibited or inhibited, do you see that they meet at the same Km value? Yeah, they meet at the same Km. So in this type of inhibition, Km remains normal. I've told you if Km remains normal, NK, it is non-competitive inhibition. Isn't that an easier way of remembering? Yeah, if Km is normal, Km, it is non-competitive inhibition. Okay. So for those of you who want to know a bit about inhibition, I'll just tell you one-liners here. Okay, just one-liners. So in competitive inhibition, which is normal? In competitive inhibition, there is normal Vmax. In non-competitive inhibition, what is normal? There is normal Km. What happens in uncompetitive inhibition? Yes, that is a Lineweaver-Burke plot or double reciprocal plot. Okay, for want of time, I'm not getting into the details. I'm just telling you the facts that you need to remember. In competitive inhibition, there is normal Vmax. In non-competitive inhibition, there is normal Km. In uncompetitive inhibition, you will have to expect parallel lines. In the line that I showed, you saw that two lines come and meet the same point, meet at the same point in the x-axis. Instead of this, if you see parallelly shifted lines, it is uncompetitive inhibition. Got it? So apart from these MCQs, these are the probable MCQs which many students did not tell me. So oxyhemoglobin dissociation curve we have already discussed. And then iron overload. I heard that there was a question on iron overload. A 10-year-old girl who was given repeated blood transfusion. What is the investigation that you will do to detect iron overload? Right? And if there was something called as liver iron concentration. Yeah, liver iron concentration, that would be the reference method for checking iron overload. I think there was, other than that, there was MRI in the liver, transferrin saturation, ferritin, right? The graph was different. Okay, but have you understood how to interpret the graph? If the graph comes and meets at the same point in the x-axis, it is non-competitive inhibition. If you see parallelly shifted lines, it is uncompetitive inhibition. If the two curves come and touch at the same point in the y-axis, if it touches at the same point, that is competitive inhibition, Gautam. Okay. Okay, so for iron overload, what is the investigation that you would do? It is liver iron concentration followed by liver biopsy, chemical estimation of iron load. Okay, and then this, I was told by few students that there was a question on enhancers. How many of you saw a question on enhancers? And I was only told 3 choices. Those 3 choices were given to me by PK. Are they specific? No question from enzyme inhibition. Which session did you attend, Mohammed? Morning or evening? Yeah, TM is melting temperature. Mohammed, did you attend the morning session or the evening session? So what are enhancers? Enhancer is nothing but a type of regulator. Okay, it's a type of regulator. And regulators, what do? They increase or decrease the level of gene expression. So regulators are of 2 types. One is enhancer, the other one is repressors. Enhancers increase the level of gene expression. Repressors decrease the level of gene expression. Now, are they specific? They are not specific at all. An enhancer is going to be common to multiple genes. So, they are not specific. Are they present only upstream? An enhancer can be present upstream, it can be present downstream, it can be present within the gene sequence. Its location is not specific. Okay, so choice A is false, choice B is false. Do they bind to co-repressors? They don't bind to co-repressors. What are repressors? Repressors decrease the level of gene expression. Enhancers increase the level of gene expression. So they do not bind to the co-repressor. So based on the three choices that were given to me, all these three choices are false. So if any one of you can tell me the fourth choice, I'll be able to tell you the right answer. Okay, so what is the fact that I've told you about enhancer? Enhancer is a type of regulator. This regulates the level of gene expression, and they are not specific. They are not specific for upstream or downstream. They can be present anywhere. They don't bind to co-repressors. They go and bind to inducers. Okay, so these are the questions that I have received so far. And if you have any more doubts in the exam that you attended today, you can ask me if it's related to biochemistry. Yeah, any other question? If you have no more questions, I'll meet you soon with a session. I think I have planned few sessions for NEET PG 2023 on live YouTube. So once I plan the session, I will all let you know. Ma'am, curves are in sigmoidal shape. Which curve? Oxyhemoglobin dissociation curve is sigmoidal. Yes. Is that your question? You mean in the image it was sigmoidal? Oxyhemoglobin dissociation curve. Was it like this? If it is like this, then the shift of the curve to the left can be either because of carbon monoxide poisoning or it can be because of fetal hemoglobin. Both are right. If it is sigmoidal, you can choose one of these two. If it was hyperbolic, it is myoglobin. The curve that is maximally shifted to the left. Yeah, the curve that is maximally shifted to the left is myoglobin. Okay. Different curve was given, different graph was given for competitive and non-competitive inhibition. That's not a worry, Dharmendra, right? Yeah, that's not a worry, Dharmendra. All that you will have to see in the graph is what has happened to Km and what has happened to Vmax. Do you understand that? If Km and Vmax, if both are altered in the same direction, if both Km and Vmax are low from the graph, then that is uncompetitive inhibition. That is how you can interpret it. Yeah, any graph can be given, any form of graph can be given, but ultimately what you're going to check is what has happened to Km after inhibition, what has happened to Vmax after inhibition. If both are low, it is uncompetitive inhibition. If it is normal Km, it is non-competitive inhibition. If it's normal Vmax, it is competitive inhibition. That is how you can interpret it. Okay. So thank you all. See you all sometime later with another session.